Good evening, everyone, and welcome to the first of this year's Oxford Botanic Garden and Arboretum Autumn Science Lectures, sponsored by Plants, People, Planet. My name is Simon Hiscock, and I'm the director of the Oxford Botanic Garden and Arboretum, and it will be my great pleasure to introduce tonight's speaker once I've gone through the usual housekeeping information. Firstly, this lecture is being recorded, but your camera and mic are turned off, so you are not being recorded. If you would like to ask a question to the speaker, then use the Q&A function at any time, and I will put these to the speaker at the end of the talk. If you need to contact us about anything technical, please use the chat function and one of the team will get back to you. The recording will be available for attendees after the event, and we will be sending you an email with instructions about how to access it. So now to tonight's speaker. I'm delighted to welcome Dr. Roberto Salguero-Gomez, who's an Associate Professor of Ecology in the Department of Biology here at Oxford. Rob's research spans many disciplines, from biochemistry and physiology to field ecology and ecological modeling, and many different organisms too, both animals and plants, as he seeks to better understand demographic aspects of an organism's life cycle. Rob is truly an international biologist. He studied environmental sciences as an undergraduate at Cadiz University in Spain, and then moved on to a PhD in ecology and evolution and biodiversity at the University of Pennsylvania. Following his PhD, he held research positions in Germany, Australia, and Ireland, before moving to the University of Sheffield in 2016 on a NERC Independent Research Fellowship. In 2017, he moved this fellowship to Oxford, where he was swiftly appointed Associate Professor, now with tenure. Rob has won numerous high-value research grants, totaling over 25 million, and published over 100 scientific papers, many in the highest impact journals, including Nature, Ecology and Evolution, Nature Communications, Science Advances, PNAS, and Ecology Letters. This is an exceptional achievement for someone who only completed their PhD in 2011, and makes Rob one of the rising stars of Oxford biology. This evening, Rob will be posing the question: have plants tapped into the fountain of eternal youth? So, Rob, I'm going to hand over to you to maybe answer that.
Thank you very much, Simon. And can I just confirm if you can hear me and see me okay?
Yeah, all good here.
Fantastic. And I'm going to share my screen. And I'm going to ask you again if you can please confirm with me whether you can see the screen.
Yeah, that's all good too, Rob.
Fantastic. And it's on the presentation, not the presenter's mode, correct?
Yes, that's right.
Fabulous. Thank you so much. So I'd like to start by thanking my host at the Oxford Botanical Garden. It's a really true pleasure to be able to interact with you. Thank you very much for allowing me to share with you some of the research that I myself and my research group at the Department of Biology have been conducting for the last 6 to 7 years. And hopefully I can convince the audience that when we try to think of humans, and we tend to think of humans as relatively long-lived creatures, and we tend to think of humans as the center of the universe to some extent, I like to pose the fact that we are neither long-lived nor exceptional in many angles. And I think that we can take a great deal of inspiration by jumping kingdoms. That's very much what I have been doing for quite a few years, and I have taken great inspiration in asking the question whether plants break all rules that we thought to be universal. senescence, as I will define in a bit, among them. And so the idea of the fountain of eternal youth, here depicted by the painter Lucas Granach the Elder, a painting from 1500s, is one whereby humans would, in an effort to remain young and not to die, would either drink from this famous fountain or bathe in it. And the mystic waters would endow them with rejuvenation or with the stopping of the biological clock. Can plants actually do that? Because we certainly, for once, humans, we cannot. We are very good, humans, obviously. We are very good at being able to guesstimate how old different individuals in human populations are. Here you have two photos of the same individual. Obviously, you can notice some key differences, and I'm sure you would be with me when I say that the photo to the left was taken longer time ago than the photo to the right. So the individual on the left is younger. There's a few key features here that give that away, one of which has to do with the amount of hair, the lack of wrinkles, the posture, maybe the amount of muscles in the individual to the left, the lack of glasses, etc., etc. As we age, we humans, we become more decrepit, we senesce. And I want to spend some time first, before I tell you about whether plants can actually tap into the fountain of eternal youth, making the case that aging is not the same thing as this interesting word that comes from the Latin, senescence, which you can vaguely think of as to become decrepit with time. So I'm going to define aging for the rest of this presentation as the mere passing of time. The clock is ticking. If you get to survive from 1 year to the next, congratulations, happy birthday, you get to blow the candles of another cake. We think of aging as the mere passing of time without any contribution to whether individuals become more fit, less fit, or remain as equally fit as they age. However, in biology, we have got a very special term for the loss of vitality in individuals in different populations as they age. Here you have got two houses. Picture the one to the left is as old as the one to the right. So they have got the same age. However, the house to the left has remained better maintained than the house to the right. So you could think about senescence in terms of the house to the right having senesced faster than the house to your left. So senescence implies the loss of vitality because maintenance has not been kept up. In biology, and more specifically in demography, we tend to think about senescence as the following pattern. Could I please ask my host to confirm if you can see my cursor moving on the screen, please?
Yep, that's clear.
Thank you. So this is a diagram that is meant to describe the typical mortality trajectory in red and fertility of a human population, a human cohort, from the age of birth until the last individual in that cohort has passed away. The dashed white line is the age of maturity, past which there's a main peak of reproduction, which then decays. As you can see, after the age of first reproduction, the risk of mortality increases, particularly rapidly late in life. This pattern of sharp declines in fertility and sharp increases in mortality is known as senescence, at least looked at from the demographic perspective. This, pun intended if I may, is a really old question. Why do we senesce? Aristotle, the philosopher, already wrote the first record that we have evidence for on some tips as to how we could actually slow down senescence. He suggested that going for long walks every day and not eating too much. So in a way, he was already suggesting dietary restriction, one of the main theories of aging. Those two activities, keeping healthy and eating healthy, could result in the prolongation of human lifespan. So this is a really old question. And it's a question that is really important for human societies, how old we get to live and under what conditions we remain healthy. That is, how long is our health span, not just the life span, will determine when we go to work for how many hours a day we work, how many days a week we work, when we retired, and many other things, such as how much money is a bank willing to give you for the first loan of your car, those calculations are based pretty much on how much life left actuarial sciences has estimated you have got in you. So senescence has got clear implications for human societies, but senescence also has got very important implications across the tree of life. Just picture what you probably had for breakfast this morning, maybe an orange juice, maybe you had some cereals, maybe if you're enjoying a nice glass of French wine. Those products come, of course, from individuals who are reproductive. And if individuals lose that vitality very quickly, then we need to think very carefully about how we exploit those resources. What's interesting about longevity and what's interesting about senescence is that it has changed very quickly in the last few hundred years. Let me take you back a few millennia to the time of 0 BC. In the time of the peak of the Roman Empire, individuals who would be the longest lived, maybe the Caesars, in the exception of an assassination, would probably reach about 40, 45 years old max. I'm 42, and hopefully, I touch on woods, I will remain fit and I will attain a long lifespan for a long time. The individual that you have got in front of you currently holds the world record Guinness for longevity. It's a French lady who passed away a few years ago by the name of Jean Calment, with a death of 122 years. Now, we have become pretty good at identifying age in human societies. And then I would put it to you, how old do you think these creatures can get to live? You have got albatrosses, the Nile crocodile, you have got elephants, and very importantly, you have got some representatives of the plant kingdom. To your top left, you have got the famous bristlecone pines. That's something that, in order to understand whether senescence is universal, first requires that we become really good at aging them. And one of the main challenges in being able to explore the question, how long lived can plants be? Do they undergo senescence or not? We must become detectives of sorts. We must become CSI detectives, whereby we have to be really good, very creative at being able to age them. Because unlike you and I, any organism across the tree of life is not born with a birth certificate that you can readily check to ask the age of the individual. There's this question about why do we senesce or why do we not senesce has attracted a lot of attention. There's been a lot of theories put forward. I'm just going to overview the main key ones, and I'll jump straight into some of the main results that prove or disprove these theories. Meta-work suggested that the reason why we senesce, the reason why we become decrepit with age, is that in simple terms, our biological machinery is imperfect, and the rate at which mutations are accumulated in our DNA is much faster than the rate at which they can be fixed by our biologies. So we senesce, we become more decrepit with age, simply because we live long. There was another theory, a really interesting theory put together by Williams, who suggested that because some genes can have more than one role, and those roles, those functions, can be split through time, certain genes exist called pleiotropy genes that natural selection could be selected for. And those genes would inevitably lead individuals to senesce. These genes would be those that have positive contributions to performance of the organism early on in life, but negative contributions to performance of the individual late in life. What matters more to natural selection is what happens early on in life, and therefore those genes that would result in organisms becoming more decrepit with time would still be selected for, and that's how senescence, Williams argued, would come to evolve. And the last of the three most commonly used one, by Kirkwood, state that organisms that have a separation between soma and germline, so the germline is the reproductive organs of an organism, the organs that are mostly focused on passing on of the genes to the next generation. And the germline is anything else in your body that is not the reproductive organs. So in species where there is a clear separation between reproductive organs and everything else, there's a suboptimal management of the investment between maintenance of the organism and the passing on of resources to the next generation. The inevitable consequence of which, this suboptimal management, is the decrepitude, a decline in vitality with age. Of course, a lot of research has been allocated to these questions now for over 100 years. And one of the main things that we know is that actually senescence does have a genetic basis. We can have different cohorts of fruit flies where we upregulate or downregulate different genes. And the longevity, as you can see here in the x-axis, the amount of days that these cohorts of fruit flies will get to live, but also the slope with which they will start to die, can be genetically manipulated. But importantly as well, the diets, the environment, the ecology of the organism is a really important source that will determine how long-lived species can get to be and how sharp the rates of mortality will be. Nonetheless, one of the founding fathers of senescence research, Hamilton, was so convinced with the idea that senescence was universal that he went on to publish in a peer-reviewed article the quote that you can read at the top. Senescence is inevitable, even in the farthest reaches of the universe. And the quote actually continues, across the most bizarre of creatures. So what he was positing was that this pattern that you can see here of sharp declines in blue of fertility with age and sharp increases in mortality with age should be universal. From lions to dandelions, from monkeys to monkey orchids, and anything in between. This tells you two things. The fact that this quote got published, it tells you that the researcher himself was very sure about that, and that at the time when this piece of research was published in 1966, the peer review community that reviewed this paper believed that that was the evidence and that that was the case, that senescence was universal. One of the limitations of that assumption, however, is that we simply didn't really have a lot of data to explore how mortality may or may not increase with age and how fertility may or may not decline with age. That is up until relatively recently. So one of the main efforts that we have been conducting in my lab is accruing, collecting, complementing, and making fully open access data across animals and, very importantly, plants. These are the compadre and compadre databases, which now allow us to actually put to the test this hypothesis of whether senescence is actually universal. And so we published a paper in Nature a few years ago where we tested this hypothesis. And what we can tell you is that modern Japanese human societies, hunter-gatherers, guppies, and albatrosses do follow this trend of sharp declines in fertility from the age at maturity until pretty much the last individual in the cohort has died, and increases in mortality. However, there are many other species that do not seem to follow that expectation, that Hamiltonian expectation. For instance, there's some trees like sequoias, some reptiles like the Nile crocodile, and even some mammals like the yellow-bellied marmots, where while the risk of mortality does increase, fertility never declines. So these are individuals that, as they grow older, in a certain way, some components of their demography get better, like a good French wine. As we walk into other taxonomic groups like brown algae and other plants, what we can see is that increases in fertility seem to be the rule, and declines in mortality are actually quite prevalent. My attempt with this figure is to break rule number one of presentations. And that is that you shouldn't have too many figures within a single figure. But allow me to explain what is going on in this figure. This figure is organized such that I'm showcasing over 300 plant species, I think it is. Each pixel, each panel is representing the demographic profile of changes in mortality in red and changes in fertility in blue from the age of maturity until the last individual in that cohort has died. And this is uniquely looking at algae, mosses, herbs, shrubs, trees, palm trees, and then for comparison, we also have got some representative of the animal kingdom, mainly birds and mammals, which is where we happen to have most of the demographic information. The pixels are organized in such a way that species to the top left are escaping from senescence, and species to the bottom right undergo very fast declines in vitality. So very fast declines in reproduction and very fast increases in mortality. What we can see actually is that across the over 300 species that are represented here, more than 50% of them do not show Hamiltonian expectations of senescence. And we can see actually a lot of greens. We can actually see quite a few purples. So all of these colors that you can see on the top correspond to plants, whereas the colors that correspond to the bottom, you can see more birds and mammals. So there seems to be certain differentiation across both kingdoms of the likelihood of undergoing senescence, with plants being less so. One of the ways in which we can think about whether different individuals within populations of given species undergo senescence is by tracking their cohorts. Picture this. Imagine that we track a cohort of individuals that all of whom have been born at the same time. And now what we're going to do is we're going to see how their survivorship changes from the time at which the first one reaches sexual maturity until the time where 99% of them have died. Naturally, you can already see that there are different types of curves. Ecologists refer to these curves as type 1, type 2, and type 3. The type 1 curve corresponds to species where mortality goes more or less flat, but as individuals become older, there's a sharp decline in survival. That is a fast increase in mortality. So type 1 corresponds to species that undergo fast senescence for mortality. The opposite can be said of species that follow the type 3 type of curve. These correspond to species that reaching maturity have got a pretty constant rate of mortality, or even a negative one, in fact. We can actually quantify these type 1, type 2, or type 3 type of mortality curves by calculating a measure of entropy, which is simply put, a transformation of the area under each of those curves. The important thing to remember here is that when that measure of entropy is lower than 1, what that means is that the species of interest is undergoing senescence. And when it's greater than 1, it is escaping from senescence. Now we can go away and apply these very basic principles of differentiating the rates of senescence as per Kivitz' entropy to various taxonomic groups within the databases that I have developed. In blue, I present you with some taxonomic classes of the animal kingdom. In green, we have got our beloved plant species. We are showcasing distributions and likelihood of undergoing senescence for mortality. If the main points are located below the line of 1, those will be species that will show senescence. And above, those will be species in taxonomic groups that manage to escape from senescence. What you can see here is that creatures like fish, birds, mollusks, insects, and mammals, as well as a significant amount of flowering plant trees, do undergo senescence. But there's quite a few exceptions. A lot of them are based in the plant kingdom, but even within the animal kingdom, corals, reptiles, and then of course you can see cycads, species in the groups of the pines and lilies, have got a propensity to escape from senescence. Now, the question that has become a primary question in senescence research really is, how do these species manage to do that? And so ecologists have gone to the 4 corners of the planet looking for bioindicators that will help us understand and predict how likely species are to live and whether or not they will undergo senescence. This research had been primarily focused on birds and mammals. And this is a really interesting paper published by a previous postdoc in my research group, Kevin Healy, where he showed a positive relationship between body mass and longevity. However, you can see some exceptions. While bigger species like the elephant live longer, you can find, for instance, species that, like bats, these triangles, given how small they are, they live much, much longer than other species of a similar body mass. The opposite can be said for birds of really heavy weights compared to other ones that do fly. Still, the fact is that even though there are some exceptions, some notable exceptions, there tends to be a positive correlation between body mass and longevity. And the thought behind that positive correlation is that when you're big, you're difficult to be eaten, or you're difficult to be herbivorized. But also when you're big, you have access to more resources. Introducing one of the longest lived species in the plant kingdom, Pando. Pando comes from the Greek, everything. Pando is in fact not just a tree, it is a forest. Pando is a specimen of quaking aspen, Populus tremuloides. It is found in the US, in Utah, and it occupies hectares and hectares of forest. All of it genetically similar. It's the same individual, in fact. Pando is currently estimated to have achieved a lifespan of over 80,000 years. Let that sink in for a second, and may we think again when we think of ourselves, humans, as extremely long-lived. But you don't really have to be extremely big or extremely tall to be long-lived. I would like to put a game to the audience today. I'm going to give you a few seconds. If you're watching this with your family, I would challenge you to guess the right age of Pinus aristata, the Rocky Mountain bristlecone pine. Your options are 106 years, 1,100 years, 2,021 years, or over 5 millennia. I'll give you 2 seconds for you guys to discuss. And the drum roll goes to, and the winner goes to option D, over 5 millennia. So contrary to Pando, bristlecone pines do not grow clonally. The reason that Pando has occupied such an extensive area of hectares is that it sends underground stems from which a new stem can grow. So the whole forest is actually a massive clone. That's one of the reasons as to why some plant organisms are believed to have been able to tap into the fountain of eternal youth. In the case of bristlecone pine, clonality cannot be the reason, as clonality does not happen. What could it be then? We'll tap into that a bit later, but I would like to leave you with a sobering thought of just how old these creatures can be. King Minis was the first pharaoh of Egypt. At the time where the individual bristlecone pine on the bottom right had achieved 100 years old, which is about the time at which juveniles come of age and start to reproduce in that species, King Minis was about to unify the Egyptian empire. In other words, there are some currently alive and kicking individuals of Pinus aristata that were very much alive and kicking and starting to reproduce by the time that the Egyptian empire wasn't even a thing. So bigger, longer lived, but also there can be some very notable exceptions. So what I'm showing you here is a publication from my research where we classified plant species according to longevity on the x-axis from short-lived to long-lived, and fertility on the y-axis from not very frequently reproductive to highly frequently reproductive. Each dot corresponds to one plant species. And the color corresponds to a rather old but very handy way of classifying plants, the Raunkier growth form classification of botany. Raunkier, a Swedish botanist, classified plants according to where their centers of growth, their shoot apical meristems, overwinter. So you have got species like helophytes and geophytes whose shoot apical meristems overwinter below water or below ground. Hemicryptophytes or cameophytes, these are also very small plants that overwinter around ground level. Epiphytes, these are plants that overwinter on top of other plants. And then you have got nano, meso, and mega phanerophytes. So these are trees. These are trees that reach up to 5 meters, 10 meters, and above in total height. So the main thing that I want you to focus on here is that brown means very tiny, green means quite tall, and the height of a plant is a good proxy for its biomass. As you now walk from left to right, you can see that there is a gradation going from brown to green, but you can also observe some exceptions. I don't know if you can see where I am putting my cursor there. That is a cameophyte, and you may be wondering, what is that tiny thing doing in a realm that is virtually occupied by very long-lived trees? Introducing the herbaceous perennial Borderia pyrenica. Borderia pyrenica is an endemic plant that lives in, as its name says, the Pyrenees between Spain and France. This species, even though quite small, has been recorded to live over 300 years. The researchers were able to follow, using some really interesting tricks, the probability of flowering, so reproduction, and the probability of survival for males and females of this species. This species, by the way, is dioecious, meaning that it has separate sexes in different individuals. And the way that they were able to ascertain the probability of flowering and survival was not by looking at the birth certificates. We are the only species with that gift, but by taking a really interesting shortcut. Every year, the tuber that is found underneath in the ground will produce a leaf. Hopefully, you can see here spirals of scarps of the leaves in previous years. In fact, one of the belief reasons as to why this species can get to live so long is that out of the 5 shoot apical meristems that it has, it only activates 1 every year, and the remaining 4 take a break. The scarves that are left after the leaf wilts at the end of the growing season allow the researchers to estimate the age and to link that age to the probability of reproduction and survival. And what you can see here is that as individuals of this species get older, there's no sign of declines in fertility or increases in mortality. On the contrary, they tend to get better. So what this is doing is allowing us to become quite creative. I'm sure that you know that in certain plant species, we can count rings, but that is not the case in every species across the tree of life, nor specifically within plants. So there's different ways in which we can actually do effectively dendroecology. On the topic of whether body size is a good proxy to longevity, we know that within the animal kingdom, there's some species that can fluctuate in size from one year to the next. Introducing the marine iguana of the Galapagos. These are endemic reptiles that live in the Galapagos, and they're known to shrink in size quite a lot. When they undergo shrinkage, this loss of body mass, is mostly associated with a decrease in resources. Decreases in resources in that part of the world are followed by a La Niña event. So what happens with this species is that a La Niña happens, there's fewer resources, the algae that they usually eat, they're smaller, there's not as many resources, and the only way that they have to get to them, to get in between the rock crevices, is by shrinking. Now, when I'm saying that they are shrinking, I don't mean that they put themselves on a diet. This is not them just not eating for 2 days. Shrinkage happens in these species by the physical resorption of calcium from their skeletons. So when you measure an individual the year prior to La Nina, from the snout to the cloaca, which is where the organism does its business, it reproduces, it exchanges other parts of its body with the environment, that distance actually shrinks because of the consumption of the skeleton the year after La Nina. And what these researchers have shown is that individuals that shrink the most have a higher rate of survival. So being able to be plastic in your size in the plant kingdom has been demonstrated to have benefits. This then led me to ask the question whether this could also happen in the plant kingdom as well. And I would like to take a second to thank Nintendo for having allowed me to take inspiration from a video game from the Mario Brothers. In this video game, for those of you who may be young enough perhaps to have played that, Mario has to save a princess. But he's got two shots. If an enemy injures him, he will not die immediately. He will shrink. If the enemy injures him again, he will be out. But the ability to shrink will allow him to remain in the system, to remain put. So I asked these questions also within the plant kingdom. And it turns out that there's a lot of species where shrinkage is associated with a prolongation of longevity commensurate to the body size of the species. This has happened in many species that you can see here. It tends to happen most frequently in deserts as well, but there's also other species in the animal kingdom where shrinkage has been associated with increases in fitness, such as the tiny little shrew that you can see on the bottom right. This happened in one of the species that I studied during my PhD a decade ago. This is Cryptantha flava. Cryptantha is from the family Boraginaceae, and it's endemic to the Great Basin Desert in Utah. Some of the research that I was really interested to conduct with this species was to ask how come that some individuals can undergo such a drastic decrease in size, and how is that related to their longevity? What you can see here when I circle the cursor around it is some leaves and flowers, but what hopefully you can also see is a section right next to the plant of dead tissue. This whole individual had over 200 rosettes, groups of leaves, and the individual drastically shrank from one year to the next, going from over 200 to about 25 leaves. The way that plants can drastically modify their size without resulting in death is by internally organizing their organs. And the reason that I can demonstrate that is by doing some really interesting dye tracking experiments. So what I can show you here is a diagram of the root system of this species. The species has got a really deep taproot, that taps into the deep layers of water in the desert, and then some lateral roots whose main role it is to quote unquote look for pockets of nitrogen. Very carefully, I dug out sections of the sand in the Great Basin Desert, and I cut one of the lateral roots, and I put that root immediately in a vial with a solution of dye with puchin, which is an extremely staining root, dye, by the way. You can only imagine, on a windy day, by the way, I would end up completely covered in red and pink. I looked like I had just come back from a rock festival or something. Anyways, what I was able to show is that the solution of dye that was fed only to a single lateral root in a small plant, in a juvenile, a plant that hadn't reproduced yet, will travel throughout the anatomy of the plant and show in all of the leaves across the above ground biomass. In contrast, when I repeated this and I did that with adults, the dyes will travel only to a subsection. So somehow, as the plant grew older, it had a way to making sure that the resources obtained by the different lateral roots were traveling only to one section. And I would argue that that was exactly how these decreases in size could happen. Now, picture what could happen if you're able to collectively shut down one of the sectors of your anatomy. And think of the benefits of doing that when the resources that come to you a given year are not enough to sustain your entire anatomy. It would inevitably follow that a strategy that can shut down different modules would allow the rest of the anatomy of the organism to live longer. How do they do that? This is a series of cross sections of the stem. Just to go back to the previous slide, the cross section was made either here or here. So at the unifying highway of the resources being taken from the taproot and the lateral root and going to the above ground biomass. And what you can see here going from A to I is a white bar right next to each cross section. The white bar, by the way, is 1 millimeter. So as you're walking from left to right and from top to bottom, you're actually going from really small cross sections of stems to rather thick ones. You can see here is that as the individual grows wider in horizontal dimensions, the inside starts to rot away and some channels start to develop that result in the physical separation of the phloem and the xylem, the vasculature, the vessels, the highways that are conducting and allowing for the water and the minerals to go from below ground to above ground. In fact, in these two last photos, what you can see is the stains of one of my vials of green dye and the fuchsin, the red dye. You can see that it's only concentrated across a specific section. So to recapitulate the first part, modularity, that is the ability to split yourself, your anatomy, can lead to a longevity that is much longer than what would correspond according to your body mass. Another thing that we have also paid particular attention to in my lab in the last 5 years is whether the environment where the species live is important. It's an important predictor of senescence, of health span, and total longevity. As I mentioned earlier in this presentation, Aristotle already pointed out to eating well, not eating too much, caloric restriction. The idea behind caloric restriction, of course, is that by not oversaturating your body with a lot of resources, the cells will be less likely to produce ROSs. When you take in resources, those resources will eventually help through chemical reactions to obtain energy. But those chemical reactions can be quite ineffective sometimes, and a byproduct of those chemical reactions is reactive oxygen species. This reactive oxygen species then can go and bind with DNA and RNA and result in the loss of functionality, therefore an increase in senescence. We of course know that around the globe, there are some habitats that have got more nutrients and others that have got fewer nutrients. And some of the research that we have published in my group recently points out to the fact that those species, both in the animal kingdom, but also in the plant kingdom, like bristlecone pines, that live in nutrient-limiting environments live longer. The species to the top right, that beautiful and highly charismatic species, by the way, made the cover of Nature a few years ago. That is the Greenland shark, and it's conservatively estimated to live up to 450 years. The commonality between where the Greenland shark lives and the bristlecone pine is that obviously they live in different environments, water and land, but both environments are quite depleted from nutrients and there's not many resources. My group and other research groups have published quite a lot at the interface between investments in survival, which will result in long longevities, and reproduction. And what we have systematically found in my group and other independent groups is that most species show a strong trade-off between growth and survival. So species that grow fast don't live long, and species that live long do so at the cost of not growing very fast. In the plant kingdom, that is particularly important because at least in trees, it's been shown that if trees grow very fast, they will succumb under their own weights because the wood that they have produced is of suboptimal quality. So this trade-off, growth and survival, can mechanically limit how long-lived species can be, but the environment where those species live will also modulate that trade-off. Species that, as much as they would like, quote unquote, cannot grow fast because they are in deserts, they're bristle confined, or in rather nutrient depleted oceans like the Greenland shark, are stuck in the strategy of growing really slow, the byproduct of which is, I would argue, attaining long longevities. When it comes to plants, and related to the first point of modularity, we can actually think of the vasculature of plants as a collection of straws, as I mentioned before, each of which can act more or less independently. In a publication published a few years ago by some members in my lab, Conor Bernard and Aldo Compagnoni, we looked at the correlation between the rate of senescence in the y-axis and the degree of modularity. That is just how split or how far away are those straws. Here, what we find is a negative correlation. Each dot corresponds to a plant species. Across the pool, the rate of senescence tends to be lower than 1, implying in this case that the species tends to escape from senescence, but that as the degree of modularity becomes higher, the rate of senescence is closer to 1. So the species have a higher tendency to escape from senescence. Again, indicating the idea that modularity is a main contributing factor in the plant kingdom to the escape of senescence. In the same publication, we also looked at whether modularity in animals, birds, mammals, reptiles, corals, sponges, would lead to the same results. And we unfortunately didn't find a very significant trend. So we know that modularity is an optimal predictor of senescence in plants, but it doesn't work in animals. In the last line of research that I would like to share with you, I'd like to put to the test some of these ideas in the field. The species with the pink flower that you can see on your screen lives across the Mediterranean basin, but I was lucky enough to be able to conduct fieldwork for the last 11 years in Montserrat Mountains National Park in Barcelona. The species is Cistus albidus, the Mediterranean rosebud. What we did with this species is we followed hundreds of individuals to which we either did nothing through time, in this case 200 individuals, with the idea that through time, each of the modules would accumulate DNA damage. Other 200 individuals, which we exposed to a resprout forced treatment, so in the second year, we cut half of their biomass, and what will happen the year after that is that they started to grow new tissue. The idea of comparing these two treatments is that by forcing the regrowth of young tissue, the rates of senescence will be lower. So the individuals will show a rejuvenation of sorts compared to those individuals that were not forced to regrow. But we also wanted to test questions related to the availability of resources. Again, this idea is based on the idea of calorie restriction, whereby if you have your diets, you don't produce as much reactive oxygen species and you live longer and don't senesce as fast. Now, modifying, halving the diet of a plant in a natural environment, which is where this fieldwork took place, is extremely difficult. We cannot simply just go into the field and vacuum out all of the phosphorus. So instead of depleting phosphorus from the soil, what we did is we added phosphorus to the soil. And here the expectation was the flip. That is, we would expect that individuals where we had added phosphorus would accumulate DNA damage at a much faster rate because they were exposed to higher resources than those in the control line. For each of the over 600 individuals of Cistus albidus, we qualified the biochemistry of them. We collected a lot of information that tells us about biochemically how healthy are they. We also quantify information about the physiology, the capability of carrying out photosynthesis at the leaf level. And ultimately, we track the demography, survival, growth, and the probability of reproduction, which in this case is quite complicated. So we quantify how many seeds were produced per capita, how many embryos were produced in each of the seeds. For each embryo, what was the viability and the germinability? And then the next year, we also track whether those seeds had recruited into the population. We have been doing this since 2014, so we have got about 10 years of data. We are also able to age the individuals, which for a subset of them, we did by destructively cutting them down and then counting rings. And we know that there's a pretty good correlation between how thick the diameter of those plants are and how old they are. So using those proxies for age, we're now in a place where we can see whether control lines and the other two treatments undergo senescence, that is, they lose vitality. They have negligible senescence, there's no change in performance, or there is negative senescence, that is, there's an increase in performance at each of the three levels: biochemical, physiological, and demographic. So what you can see here is that for the control individuals, for certain characteristics, there is a decline in vitality, but not for all. And that for certain components of the reproduction, such as the probability of reproduction and the number of seeds being produced, individuals get better with age. Compared to the resprouting treatment, when we force the regrowth of individuals, a lot of the relationships that showed senescence then flipped to constancy in that vitality, implying that we had managed to slow down senescence rates at least a little bit. And in the case of the resprouting, what we found is that we managed to turn into negative senescence quite a few of them compared to the control treatment. So the main key home messages that I would like to leave the audience with today is that senescence is not universal. Even if we humans have more than double our longevity in the last millennium, and even if the prolongation of health span is a given, many thanks to the health system and many of the things that societies do for us, senescence is not universal across the tree of life. It may be for us, but certainly not for many animals, and most certainly not for the majority, I would dare to say, of the plant kingdom. The fact that in the case of plants, there's some decoupling of the allocation of resources into growth and reproduction allows us to predict that species that live in nutrient-limited environments do live longer. And we're starting to accrue some evidence that supports that idea. And also that the chemistry, the physiology of the organisms is a really good predictor at the interface with different treatments of their performance as individuals get older. So indeed, senescence is a really complicated topic. It's a fascinating topic, and it deserves a multifaceted approach as the one that I have suggested here today. I would last like to leave you with some publications of my group. There was a special feature a few years ago that I published where we carefully re-evaluated what we knew about senescence in the plant kingdom. And some colleagues of mine and I also published a book titled The Evolution of Senescence in the Tree of Life. Now I will be more than happy to take any questions if there are any. Thank you.
Hello, Rob. Sorry about that. I was just plugging my computer in. It was running low on gas. Thank you so much for a wonderful, very fascinating talk this evening, and a big virtual round of applause for Rob. There are a number of questions that have come in, Rob. The first one from Aigan, who is in Azerbaijan and who is a PhD student, and he says, do plant hormones have any effect on plant aging?
Absolutely. Absolutely, they do have an effect. The first one that would immediately come to mind would be abscisic acid, which is known to be a main regulator of stomatoconductance. We know that when plants are exposed to drug treatments, the concentration of AAV goes up, and that allows them to regulate the resources. This is particularly a key player in desert environments, where again, longevity in the plant kingdom seems to be quite commonly expanded. So while more research is of course necessary at the interface of biomolecular biology and demography, there's some evidence that hormones, of course, do have a regulating role in the evolution of longevity in senescence.
Thank you. And another question here, do plants that spread vegetatively, like raspberries or irises, age sooner or later? So relating to your aspens, I guess, in some respects.
Mm-hmm. It depends on the unit of consideration. This is something that John Harper, the late John Harper, the founding father of modern plant population ecology, made a beautiful case to differentiate very carefully. You see, there are some plants that don't reproduce clonally, and there are quite a lot of plants that do reproduce clonally. And in the ones that do reproduce clonally, the key question is, what is the individual? So John Harper made the point of thinking about the full colony of the clone as the genet. That's a word that comes from the Latin. It refers to the fact that every section connected to it has got the same genes, genet, gene. Or if you're referring to the ramets, ramet comes also from Latin and refers to the branches. In Spanish, actually, the word rama means branch, and it comes from that word. So what we do know is that the rate of senescence of different ramets, that is independent clones within the full colony, can differ quite a lot. But the senescence across the full colony, whether it's happening in one ramet or another, can be attenuated by the performance of every other connector to the full genet. So the question is not so much whether senescence can happen within the colony, but whether the senescence rates are fast enough to upscale and occur at the same time across the whole genet. And what we see in things like Pando or Laria tridentata, what's the common name of Laria tridentata? Crassiote bush in the US as well, another clone, is that they don't. And in fact, by the way, Laria tridentata is thought to live up to 8,000 years. Another world record Guinness of longevity.
Yes, and relating to this, this asexuality that is really clonal reproduction, but there are other forms of asexuality in plant reproduction, like apomixis, for instance, the production of seeds without sex. Is there any data on whether plants that are predominantly asexually apomictic live longer than plants that are predominantly sexual?
That is such a lovely question. That is such a good question. And I hope that if somebody in the audience or you, Simon, knows the answer to this, you please share that with me.
No,
I don't think the community knows, the research community, to my knowledge, to my best understanding, we don't know the answer to that question.
No, I certainly don't. I wasn't trying to catch you out in any way. I just wondered if you'd picked up any research on this, because that does seem an important question.
Yep, absolutely.
And here's another one from the audience. Is the ability to reproduce the sole criterion for senescence? If so, what about individuals that cannot reproduce? If not, and there are other criteria as well, why do organisms finally die? Brackets regarding the ones who actually die naturally if they do not undergo some form of senescence.
Okay, so I might, Simon, I might have to ask you to remind me some of the questions. Let me take it by parts because I feel like there's 3 or 4 really interesting questions there. So the first one is a decline in reproduction the only criterion to quantify senescence? And the answer to that is not at 2 levels, in fact. So we can quantify senescence in plants, in animals, in microbes, according to multiple levels of biological organization. We can quantify whether the vitality of the genes is declining or not. We can quantify, so that's the genetic level, right? We can quantify whether the performance of the tissue is declining or not. That will be the histological level. We can look at the same thing with the organs, so that will be physiological. We can look at the individual level, upscaling to populations. So that's the level at which some of the research that I presented today has focused. And within the demographic level, then you have got 2 components. Reproduction is one. So how much, how often, and for how long do you reproduce? But the other one, which is critical, is how much you invest in survival. So reproduction and survival are really the building blocks of what natural selection optimizes: fitness. So when we're thinking about whether a species undergoes senescence or not at the demographic level, we evaluate the risk of mortality through age and how the reproductive profile changes through age. Now, I think, and Simon, please correct me, there was a question there relating to individuals that no longer reproduce. Vaguely remember, please correct me.
Yes, are there other criteria, other characteristics that contribute, I suppose, to their death?
Yeah, so within the pattern that this member of the audience mentioned before, in the individuals that lose reproductive contributions to the population, one would think would be immediately wiped out by natural selection.
Mm-hmm.
The way that natural selection operates is by maximizing the tenet of the struggle of the fittest, as Darwin himself would say. Then you can think, okay, so if individuals stop reproducing, why does natural selection, quote unquote, allow them to remain in the population? That is the hallmark of our demography. If you think about how narrow our window of reproduction is compared to how long-lived we can be, right? We maybe stop reproducing around age 40, maybe even before, it depends on the country, of course, and we can get to live double that, right? So what's going on there? So in the case of species that have got what we call post-reproductive windows, that is a long longevity after they stop reproducing, one of the reasons why that is kind of like allowed, quote unquote, by natural selection, is that we have got other ways of contributing to the population than via direct reproduction. And that is what the grandmother hypothesis states. So when you reproduce, you're contributing directly to the population, and when you stop reproducing, you still continue to contribute to the population because you literally look after your grandchildren.
Thank you. Now, here's one. How do you think plants will adapt with the climate extremes we now see, and what plants should we look at planting for the future? So
That is such a good question.
Fairly general question. Yes, it's a very...
No, but this is a fantastic. I would say this is the not the one, but maybe the $10 million question or £10 million question, right? And this is actually a question that keeps me occupied. So a lot of the research that we do in my group has to do with the mechanisms by which species, plants in other kingdoms, escape from senescence, and the other one has to do with ecological forecast. So we use mathematical models coupled with demographic information to predict who is going to win and who is going to lose before climate change. And the interesting result at the interface between what I just mentioned is that species that have got long generation times, long longevities, species that postpone the first reproductive event, there's combines among them, right? I mentioned that they usually wait up until 100 years before they mature. our research shows are more vulnerable, are more sensitive to increases in extreme weather events. So what this means is that on the one hand, there are some species that are evolving longer lifespans, but that in itself might be penalized by things that are happening climatologically speaking.
Thank you. And now another question. When plants are infected by a pathogen, there is a trade-off where the plant stops growth to resist infection. Would immune signaling therefore have an effect on the longevity of plants?
This is a fantastic question. And in fact, it gives me the opportunity to add more complications to this. I think that yes will be the short answer to the question. So it would definitely affect the rates of longevity and senescence. But this also gives me the opportunity to open up really briefly, Simon, if you allow me, a very active and current debate in the field of senescence. And that is, can we actually study senescence in the wild? Does it make sense to follow longevity and senescence rates of plants, animals, microbes in the wild, being that things like pathogen attacks, a thunder, a fire, predators can happen, right? And so there's two schools of thought in senescence research. Those who think or strongly posit that senescence can only be examined carefully in petri dishes in the lab, under very carefully controlled conditions. And those who, I must admit, I belong to the second camp, by the way, who realize that senescence, longevity, these are key demographic properties that natural selection acts on in the wild. And so if you're likely to be attacked by a pathogen, natural selection will be doing its job to it as well. So I think that this question really opens up the realization that for quite a while, the study of plants, for instance, right, which was very difficult to do in greenhouses, was neglected. And it's only been relatively recently that we've actually started to look into the wild, which is what I would argue we need more research.
Yeah, and your study of Cistus albidus is terrific in that respect. So that's I hope many more years of research there. In you mentioning microbes, you haven't mentioned fungi at all, and I know some of these huge sort of fairy rings that are supposed to exist, well, or do exist in parts of the world, are a bit like your aspens in many respects. Could you say anything about fungi in this sphere of senescence and longevity?
I will say a few things about that briefly. The first of which is not much is known in regards to whether fungi do undergo senescence or not. Partly because it's quite complicated to identify what an individual is. Even in a hyphal networks, which are, you can think of hyphal networks as the roots that connect the different individuals within a given species of fungus, right? Hyphae can have more than one nucleus. So what is the individual? That's the first question. The second question is that a lot of these fungal networks happen below ground. And to do a careful long-term monitoring efforts, we need to observe them. By observing them below ground with the current technology, we would be disrupting them and therefore affecting their mortality rates, probably. I mean, likely there's new technologies that allow us to scan into the soil. Very expensive, and I look forward to those prices going down. But I must admit, the final thing that I will mention in this department is that I think that thinking about senescence, thinking about longevity by kingdoms has historically hurt the field. I think that animal demographers, within which human demographers are the ones that have done more work, could definitely benefit from looking at what plant researchers and fungal researchers and microbial researchers are doing, and vice versa. And the modularity that many fungi have is a copycat of what I have presented today in the plant kingdom. So many of the things that I have discussed with you guys today, I'm pretty sure would also apply to fungi.
I'm sure you're right, yes. So I think, bearing in mind the time, we'll just have time for one more question. And here we go. For organisms that do not undergo senescence, what is considered the primary reason for their eventual natural death?
Stochasticity. Environmental stochasticity. A simple way to put this would be, hopefully not, but you leave your house and you get run over by a car. Just like that, stochastic events, that random effects can happen in a human population. Things like that can also happen across the tree of life. A fire, a thunder, a massive cattle that steps on a rosette plant, stochasticity will wipe you out.
Yes, right. That's a good one to end on. Anyway, thank you so much, Rob, and thank you from our audience, even though we're not all able to clap, but that's been a really thought-provoking talk this evening, and a great way to kick off the autumn science lectures at the Botanic Garden and Arboretum. So thanks, Rob. And just before I go, I'd like to remind everyone appearing on the screen now is that we have more lectures to come in this series. So do please check out our website for the timings of the remaining lectures. And if you do have a minute, those of you that are still around, or those of you that then tune in and watch this at your own leisure, please could you complete our survey to give us your feedback by scanning the QR code on the screen now. That will help us make these lectures even more better if you want them to be, or hear how you are enjoying them. So please do complete that if you can. And thank you all for attending, and I'll look forward to seeing you next week when we have Professor Baroness Kathy Willis with us. So good night, everyone, and see you all again soon. And thanks, Rob, again. Bye for now.